From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on January 11th, 2024. Hello, Micronauts, and welcome again to today's Quality Quorum. We apologize for having a bit of a delay as we juggle various aspects of Microbe TV, and we're glad to be back. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 24th episode of Matters Microbial. As always, I continue to be genuinely grateful for every listener, viewer, and commenter. First, a valued member of my Wunderkammer. I'm a first-generation to college kind of guy, and I was born in Compton, California in a low-income family. Growing up in North Long Beach, California, I had utterly no concept of what it was like to be a scientist until I went to UCLA as an undergraduate and Stanford University for my PhD. As you might guess, I never really fit in. To be honest, the folks at Stanford were awfully smart and awfully self-assured. I didn't fit in. There was a person in my lab while a PhD student, Philippa Webster, and she went out of her way to be kind to me to make me feel like I was valued and included. I've never forgotten that. After all, I was kind of a work in progress in my 20s. Philippa took some time off from her PhD program to work at the Hansen Institute in Ethiopia. Then and now, the Institute works tirelessly to reduce the impact of leprosy in Africa. When Philippa returned, she brought me a beautiful wall hanging shown here. The bell at the bottom, she told me, had been blessed by a Coptic priest to bring me luck, and you can bet I ring it often, and I think of Philippa every single time. I put links to the Hansen Institute in the show notes, as well as some links about the bacterial disease leprosy. Today's topic of quorum sensing is particularly relevant to our quality quorum, as you might imagine. Many types of bacteria can monitor their own population levels, and sometimes the levels of other bacteria, and express different genes as a result. It turns out that this ability is very common in the microbial world. Bacteria can secrete a small molecule, which in general we can call a quorum-sensing molecule. The concentration of this molecule is an indicator of population density, and many microbes carry out group behaviors controlled by this. One of the first bacterial systems in which quorum sensing was studied is my favorite, bacterial bioluminescence in marine bacteria. As you see this glowing incubator, these bacteria at a high population density emit beautiful blue-green light. Always brings me happiness. And I often use this kind of bacteria in my classes to promote a connection between science and art. Seen here, for example, is a Lux portrait of one of the most important and impressive figures in the study of bioluminescence, Dr. Bonnie Bassler of Princeton University, who has a connection with our guest today. My wife, Dr. Jennifer Quinn, painted with these bacteria on a large Petri plate to create this lovely image. As you may know, I have put on many Lux art workshops at other institutions, and as you can see here, the Lux art that students make can be amazing and intricate. Even my first-year biology students at the University of Puget Sound can create wonderful Lux art, seen here. I even take photographs of my Doc Martian undergraduate research students illuminated by bacterial light. But quorum sensing isn't just about the production of light. The bacterium Chromobacterium makes a purple pigment called violacine. Look at those single colonies. It turns out that the ability to make this pigment is under quorum sensing control. That is, the pigment is only made when the bacteria are at high population densities. If you mutationally remove the ability of Chromobacterium to make the molecule that allows it to know it's at a high population density, it no longer makes the pigment. But what you can do, and I show it here, is I use a mutated strain of Chromobacterium to look for certain types of quorum-sensing molecules. In the case of this image, the Erwinia bacteria are making quorum-sensing molecules, and those molecules are telling the Chromobacterium nearby to make the purple pigment. Now, the process of quorum sensing and related areas of socio-microbiology are very important to me as a central paradigm in biology, and it's a very relevant topic. A number of bacterial diseases use quorum sensing to have pathogenic effects. That is, disease can be a group behavior of bacteria. And I have to say, we're very fortunate today to welcome Dr. Julia Van Kessel of Indiana University, told you I'd get it right, to discuss her path in science and her research linking quorum sensing to some kinds of disease. Welcome to our Quality Quorum, Julia. It's truly great to see you today.
Thanks for having me, Mark. It's a pleasure to be here.
I think the first thing that I like to do with our visitors, Julia, if you don't mind, is talk a little bit about your path, what brought you to where you are.
Sure. I'm happy to talk about it. So I grew up in New York. I actually was born in Connecticut and moved to a tiny little town called Millbrook, which is very close to Poughkeepsie. Some people have heard of that one. So I'm the oldest of three, and I have, as you know, a PhD in microbiology. My middle sister has a vet tech degree, and my youngest sister has a PhD also in microbiology. So that is obviously part of our family genetics, or at least we think so. I was always very interested in science as a young one, even in high school. I thought I would go to med school like many of us do, and it turns out I just was meant to do research. And the way I found this out is that when I was an undergraduate, I went to Utica College, and at that time it was Utica College of Syracuse University. It's now Utica University, actually. And I started getting into labs, so cell biology, molecular biology labs. And the professor that taught them, Dr. Larry Aronson, just really had a way with teaching, much like yourself, Mark. He just made me passionate about research. And I was lucky enough to be able to do research in his lab, starting in my sophomore year. And the minute I started, I just got hooked on it and I couldn't stop. And I have been doing research ever since.
Larry is an awesome person.
He is an awesome person. We call him Doc for short, much like yourself.
I had no idea. Mm-hmm. Do you know, by the way, why I go by Doc?
I don't.
So I used to work at an institution that had a professor named Martin Morton and another named Gary Martin.
Oh, my.
And my wife made me a button that said, "I'm not that Martin." And so I thought it was funny to go by Doc Martin as a result. Also, I ended up wearing a lot of the boots.
Well, I mean, why not? When in Rome.
Yeah, when in Rome, wear Doc Martens.
There you go.
So you have one other thing about your background. Would you mind talking about your postdoctoral work? And by the way, I am certain, the more I listen to you, that you were one of the people I talked to at a microbe many years ago.
It's very possible. I feel like you and I have been at the same microbe meetings for years. In fact, I went to ASM before it was microbe. I went in my junior year of college, so 2002. There we go. So over 20 years ago. Right. So after I went to Utica, I went and did my PhD at the University of Pittsburgh, where I was very lucky to be in the lab of Dr. Graham Hatfull. And I studied mycobacteria and bacteriophages, and it was a wonderful experience. I loved grad school. Wish I could go back. And actually, it's kind of a link to what you were talking about in the beginning, right? So leprosy is caused by Mycobacterium leprae. So there's a little connection there too. After which, I decided to apply to the lab of Dr. Bonnie Bassler for my postdoctoral studies. I had seen Bonnie, speaking of ASM, at the general meeting one year, and I was hooked on the concept of quorum sensing and on her spectacular teaching of the science during her talk, and I just couldn't imagine wanting to be anywhere else for a postdoc. So I wrote to her and applied. And lucky me, I got in.
She remains a force of nature.
As always, yes, she is. A very funny, wise, and strong force of nature.
Mm-hmm. So you got your kind of introduction to the idea of quorum sensing sociomicrobiology from your postdoctoral stint. So what did this develop into your research program in Bloomington?
That's a great question. I like to think, one of the last postdocs that Bonnie had that really focused solely on Vibrios. And if you look at some of her crew now, a lot of them are looking at Pseudomonas. Many of them that even started with Vibrio have moved on to other bugs. I was one of the last to really love and dive into the genetics of her original bacterium, which is Vibrio harveyi. And it's still my favorite bug to work on. But I think I am kind of one of the more rare people that has stuck with that from her lab. And the reason I chose to do that is that, and Bonnie and I joke about this, she cares very much about a lot of the biology of quorum signaling. So the molecules, how they're made, how they're sensed, how that's integrated, the genes that are impacted. But my focus has actually kind of been to ignore the top part of that cascade and really focus on how those genes impact bacteria in their environment. And for that reason, I think actually she and I synergize really well. And even the ongoing research in her lab in Vibrio, it works well with what we're doing in our group.
It's really quite remarkable stuff. And you know, there's a saying, I don't know if you've ever heard it, that science progresses by, 3 different stages. One is that's a silly idea, it's a waste of time. The second stage is it might work a little bit in the lab, but it's not relevant to nature. And the third level is I said it was a good idea all along. But all of the people with training in bioluminescence, and Vibrio harveyi is an old friend of mine as well, I have to tell you, there's something about having worked in that field that makes you very, very aware of connections outside the phenotype being studied. I, for example, was completely unsurprised when I learned that other types of quorum sensing molecules could impact how Vibrio harveyi did things.
I completely agree. I think we, all of us, share this love of that beautiful blue glowing culture. I, for one, I just can't get enough of it. We love to play with it. We do Lux art, not nearly to your level of sophistication, but we have fun with it too. And even when I was a postdoc, I loved to grow my bacteria in a flask. And I feel like that's just a connection we all have. But the interesting thing, at least to me, is that in harveyi, unlike in Vibrio fischeri, we don't really know what the function of bioluminescence is. There are some theories and ideas. Whereas in Vibrio fischeri, which is also a wonderful model for studying quorum sensing, we have a lot better idea of what that bioluminescence phenotype affords for its symbiosis with the squid. So I think, to your point, Mark, that if we love our bioluminescence, but we also recognize that there are many other phenotypes that are intricately controlled by quorum sensing, that we probably can imagine more relevant functions towards pathogenesis or microbial community formation or competition.
Just group behaviors.
Absolutely.
It's so easy. It's so simple to think that they're just individual cells booking around inside of a 2059 culture tube or forming a colony on a plate. Mm-hmm. But that's not where they are in nature at all.
Absolutely.
And so for me, I always suspected that there was this whole emergent property of microbes in groups. Now, it's not that I'm like super smart or anything. A lot of wonderful people had that idea. But I'm not surprised to have learned that being the case.
I think you and I are just standing on the shoulders of giants like Bonnie Bassler, Pete Greenberg, and many others. I think, but I still think that there's a lot to learn. And even I think they might imagine that we're still learning a lot as we go.
They have much to be, we're learning a lot, and the microbes still have a lot to teach us.
For sure.
So talk a little bit about the work that you do in your lab.
Absolutely. We started about 10 years ago, actually. And I had a tiny little lab, as many of us do, and we start, and I would like to think kind of just carried on a lot of the experiments I did as a postdoc, which were really focused very heavily on the biochemistry of the master regulator in the quorum sensing circuit. I won't bore you with too many of the details, but I was really invested in understanding this protein that is in every Vibrio, in all of the quorum sensing circuits, and it controls all of the behaviors that they regulate. So I thought it was really interesting. And in the beginning of my time at Indiana, I was actually in a biochemistry department. And I find it very important to know that I was actually a non-tenure track faculty member. I started with $20,000. And I think that's really important because I think a lot of our students and trainees feel that there's these really defined paths that get you to where you want to be. And I just think I've had a really amazing time in a different journey than the standard. So anyway, we started in a biochemistry department next to a structural biology lab, and it changed the way I did science for a while. So we did a lot of biochemical work on Luxar, our favorite protein. But I was then hired into the biology department as a tenure track assistant professor, and I felt like I had some freedom and money to explore some new paths, right? So we really have branched out quite a bit. We now have many Vibrio species in our lab that we study. We think about them and their interactions with their hosts and the different ways that quorum sensing has evolved in these different species. So things like Vibrio harvii are still a staple for us, but we also talk about and study Vibrio vulnificus, a huge human pathogen that is unfortunately becoming more prevalent in disease. Vibrio coralliilyticus, which infects corals and kills many corals around the world. And then even some additional ones that are non-pathogenic, like Vibrio nitrogens, and then some others along the way. So we've really kind of spread out, and we still think very hard about biochemical functions of proteins, but we also do a lot of genetics and structural biology and, believe it or not, host infections now.
What systems are you using to look at infections?
Great question. So we were funded last year by the NSF and BSF to develop a coral research facility in collaboration with Blake Ushijima at UNCW and Dora Solomon at Tel Aviv. We have funds and have started a coral research facility in our department. And that affords us this really unique opportunity to grow corals. I've hired a marine biologist who knows how to do that. And we study both the Vibrio and the coral and their interaction. And of course, we started with quorum sensing, right? So that paper is being typed as we speak, and we've now got a pretty nice understanding of how quorum sensing works in that Vibrio, the coralliilyticus species.
Julia, because quorum sensing is so central to all of biology, not just microbiology, and certainly not just to this particular session, do you think you could go step by step through an example of quorum sensing so that the viewers could get some idea of the process?
Absolutely. I'd be happy to. So, I'm a Vibrio biologist, and I think very centrically about Vibrios. So I'll give you an example about my favorite organism. So Vibrios are marine organisms. They live ubiquitously in the oceans. They exist eating chitin, or a massive carbon source in the ocean, right? So they can live off of that planktonically, just swimming around. But the way I think of this is if we start with a single cell, okay, just swimming around in the ocean's a big place. So these molecules, you call them quorum sensing molecules. We sometimes call them autoinducers, pheromones, whatever you want to call them. Even if the cell is making these molecules, they're going to kind of diffuse away in that big ocean environment. But let's pretend that swimming around cell were to come across a yummy little crab shell, for example, and sit down on that crab shell and grow and divide as cells do. Over time, if it's still making those compounds, the more cells that are made, the more compounds are made, and the local concentration of those compounds will increase. probably because that bacterium has started to make a biofilm as well. And so that group of cells has this kind of nice environment where they're kind of contained and the molecules are locally concentrated. And as it grows and grows, more molecules are made, and that behavior is going to change because the quorum sensing molecules are at a high concentration. They've reached quorum. And when they reach quorum, they change their behavior through this signaling process. So if we step back for a second, let's go back to where it's a low cell density bacterium. I kind of have this like cartoony idea in my head about how the signaling can work. And some Vibrios have a couple sensors, some have 7. But for example, if this bacterium is trying to sense different molecules from itself, from other Vibrios, from other organisms, all of that is being listened to by these receptors. When there's not other cells around, those receptors are not binding anything, and those group behaviors are all shut off. But then if you switch back to them being in the biofilm on top of the crab and they're all happy and there's a million of them there's lots of molecules coming in and out of the cytoplasm, and they can bind to these molecule receptors. They're very specific on the cell surface. And when they do that, they elicit a signal transduction cascade, and that funnels down. I'm skipping a lot of steps in this, and it funnels down into our favorite protein, LuxR, and it turns on all those group behaviors, the biofilms, the competence to take up DNA, the type 6 secretion, the type 3 secretion, and of course, bioluminescence.
It's interesting to think about having a coral facility in Bloomington, Indiana.
Isn't it?
But I mean, I think so many people who enjoy saltwater aquaria have developed a lot of, kind of the concepts of how best to grow them. So it's not necessarily a surprise. I do want to take a moment to point out you have a beautiful illustration from your website about all the different things that quorum sensing can do, which I don't know who did the art, but you need to go give them some extra money now. And if it's you, go have a couple drinks.
That's kind
of the point that I'm getting. The point that I'm getting at is this idea of microbes doing things as a group, to have a group behavior, is something that a lot of people have trouble with because they're so used to thinking about single colonies and plates, single bacteria seen under the microscope, though with these eyes, I'm not a good microscopist at all. But as you can see, this idea that you learned, our old friend LuxR, which is kind of like the central playa, PLYA, involving the kind of the circuit to monitor population density, can be involved in all kinds of things that people hadn't expected. And even outside the gram negatives, there are lots of other bacteria that use similar sorts of ideas. Gram positives, for example, use short peptides as quorum sensing molecules. So this idea is so critical, and it's the kind of thing that you know, and I know, and I am darn sure that all of my first-year biology students know it too.
I'm so glad that you're teaching it to them. I think it's a wonderful concept to bring in that early. And certainly we do know now, we know so much more about what quorum sensing controls. And like you said, it's diverse. And the ways they communicate, whether it's a small molecule or a peptide or other funky things, they're very diverse too. But in the end, they do all control a lot of very similar behaviors actually that are necessary for disease. So across the board, things like across the board, things like biofilm formation, toxin secretion, ways to compete with other bacteria or other eukaryotes in their environment. These are highly prevalent and very common across the bacterial world.
And this idea of competition, there's a Lynn Margulis kind of view that everything is about symbiosis and getting along, and I appreciate that. No, there's some truth to that.
Sure.
But there's also almost a martial look because everybody's fighting for position and space and nutrients. And I have a saying that the hand of Darwin is on all that lives, and that's really a good way to look at it. So this idea of having these natural switchblade knives, for lack of a better term, and boy, people who study type 6 are cringing right now, but that's not the first time people have cringed over things I say. In any event, this idea of them being able to compete is once again part of this concept, sociomicrobiology. I think Pete Greenberg was the first person to coin that term,
I think you're right.
but I'm not sure.
I'm not
Yeah.
100% sure, but I know he has used it quite a bit, and his trainee Josie Chandler also uses it quite a bit. My guess is it's from Pete.
Yeah. So this is wonderful. So you have a group that's looking at this quorum sensing effect that causes a negative effect on a eukaryote, the coral. How does it go about killing the coral?
That's the goal of the grants. We know that it produces proteins that could also, not in addition to the type 6 secretion, just in a separate way. We know it secretes other enzymes that could damage the host. Vibrios are very well known for their arsenal of toxins and siderophores, proteases, elastases, biofilm components, you name it. So it wouldn't be surprising if Vibrio coralliilyticus had a suite of these in addition to its type 6 secretion that attacked the coral. And we are trying to figure out, right, as the Vibrio gets into the coral, what happens first, and then how does it get in, how does it stay, and how does it cause disease? And so I think that's where we're heading.
So that's one of your research groups for which you were funded. Huzzah, hooray.
Thanks.
Right? What other types of things are you looking at in the laboratory?
So I'm also funded by the NIH via a Mira Award, and that is a very broad, lovely grant, actually, because it gives us the flexibility to try new things. We do a lot of investigation with quorum sensing in different isolates of the same species, trying to understand how quorum sensing circuits may be evolving in different conditions. We think about how different environmental conditions influence quorum sensing and maybe vice versa. So things like nutrients that are around or other environmental signals. And then in addition, we've started using baby brine shrimp as another host infection system, which is actually pretty established in the Vibrio world, but they're pretty fun to play with. And there's, that's been a lot of fun. And I think then the third part that's been pretty big for our group is that we've developed a whole panel of compounds that block these proteins, these Luxar proteins in all of the Vibrios that we've tested, actually. With the exception of Vibrio cholera, I'll just put a pin in that one. But all the other Vibrios in our lab, we have this really nice series of compounds, and this has been in collaboration with Laura Brown at IU with me. And one of my favorite things about that is that these were all compounds that were synthesized and tested by Laura Brown's organic chemistry students. So it's a really wonderful teaching project. It's a cure, much like I know you have a cure as well. It's a course-based undergraduate research experience, and she and I work on it every year. We've been doing this for 7 or 8 years now, and we've got hundreds of molecules and many really good inhibitors.
And it's interesting because if you think about it, I mean, there's the Red Queen, the idea from the Red Queen from Lewis Carroll, running ever faster to stay in the same place. So the idea is if you can find this magical Achilles heel, boy, I'm using different metaphors, different Achilles heel of quorum sensing, I almost promise you there's an attempt that evolution will make to get around that.
Absolutely.
And that's why several bacteria, including, unless I'm mistaken, Vibrio harveyi, have multiple quorum sensing systems.
They do. And I think it's an interesting, what you're talking about is interfering with one of the signals, whereas the others are still intact. And for sure, that's a strategy that you could imagine being selected for over time. But what's unique about our Vibrios is that all of those different signals funnel into one protein. And that's the one we have the inhibitor for. So, a question I often get, which you kind of alluded to already, is, well, if you give them this inhibitor, they're going to evolve around it. And actually, that's a question we're very interested in asking, and mechanistically and as like a resistance strategy. So I have a student that's studying that as well. So it's an interesting concept. We use these compounds. Sure, we want to think about them as maybe being therapeutic, but they are really great tools for studying evolution of quorum sensing resistance, and evolution of quorum sensing circuits in fitness and fitness landscapes, right?
So the question that I have, and it's one that's always been with me, is the cross interactivity of quorum sensing molecules to different quorum sensing organisms. And it's kind of like a Tower of Babel in a way. Is there one language, the Esperanto of the microbial world, or are there many different languages?
Great question. And I for sure have always loved this discussion about quorum sensing. So Bonnie's lab has studied this quite a bit, as have many others, which is why we do call her the bacterial whisperer, because they do speak different languages. The way she's always taught it is that there are bacteria that have their own signal that is not made by other species, even in the same genus. And that's like their own language that they're talking to their own siblings. There are, even within Vibrios, there are molecules that are made by multiple Vibrios, but not made by, let's say, other organisms that would be nearby, like other microbes in the marine environment. And then there's this really key interspecies signal that Bonnie discovered. It is called autoinducer 2, and it is a byproduct of a metabolic pathway that half of all bacteria encode, and it's part of the methionine synthesis pathway. And because that is so inherently important in metabolism, that molecule is made by many, many bacteria and is therefore the same compound for all these bacteria, and then turns into an interspecies signal that all of them can recognize. But it's really interesting because the strongest signals, the ones that elicit the strongest effects, tend to be the ones that are intraspecies, or only within their own little family. So they can listen to other cells, but they respond the strongest to their own language. And I think that's been really interesting. And it's something that we often integrate in our studies to try to understand the genes downstream and what the strength is of the effect of the different signals. So for sure, they can speak other languages, but they're only listening to themselves in some cases.
Well, they're polyglots. They're multilingual.
There you go.
You've given me new things to teach now. It's interesting when I talk about quorum sensing with my micronauts, my microbiology students, I talk about responses that can be made to interfere with quorum sensing, quorum silencing. Mm-hmm. One idea, of course, is to physically consume the quorum sensing molecule, which amuses me no end. The other is to make a competitor to that, mm-hmm. which makes perfect sense. And I believe there are some other approaches as well, but the whole idea that I like is this idea of groups of organisms fighting together. What's cool in what you're saying is, again, there's an Achilles heel, the Luxar gene. Mm-hmm. The Luxar protein. Mm-hmm. Pardon me. I reveal my background. So in any event, so with your cure that you're doing with students, mm-hmm. I'm presuming what you're doing are looking for inhibitors from nature that seem to block an easy to measure phenotype involving quorum sensing.
Almost exactly right, but not natural compounds. We are these are all synthetic. We don't have any knowledge that these particular groups are found naturally. But yes, it's an easy to read phenotype, and I bet you could guess what it is.
Light.
You got it. So what's nice is actually we can screen all these compounds in E. coli, and we can actually even move the light protein mm-hmm. genes into E. coli. So everything's BSL-1, and it's very easy and robust to manipulate in the lab. These are OChem students, not the best micropipetters you've ever seen, so we need to keep it very robust and rigorous for their assays. But it works, and they have isolated numerous inhibitors that we hadn't had before.
And let me emphasize for the listeners and viewers that what this says is that undergraduate students can participate in a process that could conceivably have medical relevance, just as you kind of did with Graham Hatfull, right?
Yes.
And this kind of thing, often students will tell me there's no way that I could do research that matters. In fact, I've been known to brag that nothing that I've done has ever benefited humanity. So I view that as kind of a badge of honor with me. But more to the point, you can tell students it really does matter what they do. And this is fundamentally different than the kinds of labs that we often do where there is a series of exercises and observations. I'm not being negative about people who do that, please. So, I look forward to your mail, as the saying goes. But honestly, nothing succeeds like the look on a student's face when they found something they found that has this effect. Would you agree?
Absolutely. I love teaching the OChem students, my section of it. Although at this point, my chemist collaborator, Laura, can do all of it. She's learned all the microbiology as well. But it's always really fun to teach them how to use microbiology tools, like a pipette, a serological pipetter, the automated ones that we have now, that not like the old fashioned kind. They love those. They love the plate reader. They love, micropipetters and plastic tips and shaking culture incubators. And it's, I think, a very different world to take the chemists out of the chemistry building and bring them over to the biology building. And then she's been teaching them modeling of structures with their compounds too. So they really have this nice kind of iterative and multidisciplinary experience, and they get it in their freshman year.
Wow.
Yeah.
That's really quite amazing stuff.
We love it.
So now we've talked a little bit about your work with, I'm not going to say the name of the Vibrio that causes difficulties with quorum, because I will trip. But that was fun to hear about. We talked about your cure, where you're looking for inhibitors of quorum sensing. Are there other projects? Because I've been to your website. Are there other projects that you're working on as well?
I think that, they're generally always a new thing or two. So I have a student that is kind of in a reductionist way thinking about type 3 secretion in Vibrios, which is another virulence factor. That's a fun one. I have another student that is very interested in connecting to, I think I mentioned the nutrients aspect of it. So I have a couple people thinking about it. And we have a really nice story we're building on how quorum sensing actually controls key metabolic pathway in two metabolic pathways in the cell. And so it, a little more, still reductionist, but still, I think, important and teaching us new things.
And I want to emphasize again, if you go to UCLA as I did, and you think you want to work in a lab, and you're working with postdoctoral scholars and graduate students, and you have a tiny piece of that particular work, and that's valuable. And that was my first publication on human DNA a million years ago. But the thing that I want to say about this is that this is something that a student can kind of take in and say, this is my work. I did this, and I'm proud of it. And I'm very excited to hear that you're having such success with it.
I think that we have a nice way of doing both basic biology, but connecting it to translation.
Yes.
I am a huge fan of basic biology. I've always been a basic biologist, but I do recognize that desire that many of our trainees have to connect it to the real world and to go home and tell their family why it's important and what they're doing. We have a lot of students like that, and I don't fault them for it. I think it's important to think about things broadly. But I do also remind them that many of our most amazing discoveries have come from basic biology research and in microbiology nonetheless. So I think it's a good mix. It's a good lesson to learn both sides of it.
Do you know who Isaac Asimov was?
Yes.
So I don't know how much of a science fiction fan you are, so excuse me.
My dad read Isaac Asimov always.
So there's a wonderful saying that he had that I really like, because he was a good science popularizer. Mm-hmm. And what Asimov said is that science doesn't progress, and here he's being clever, by a damp Greek emerging from the bathtub shouting "Eureka." Right? Science actually advances when someone looks carefully and says, that looks weird. And that's 100% true.
It is.
For all of our talk about very expensive pieces of equipment, the most important one is between your ears. Mm-hmm. not necessarily mine, but certainly with the students. And one of the challenges that I have is to teach them that their observational skills are critical. When they notice things, it is worth bringing up. And that's why I'm so excited by the cure that you've set up, especially because it's involving lots of different people from different areas, because I think that's critical.
I do too. I love that they come in with a different perspective. So the chemistry undergrads, many of them I end up having to do full-time research in my lab. And they do have a different way of looking at things and asking different questions. And it's incredibly valuable to have their expertise with us. So I absolutely agree.
Two things that occurred to me as you're speaking. Would you mind?
Of course.
I seem to remember, and if I'm wrong, I apologize, that Vibrio campbelli is associated with some types of shrimp diseases, which you find in aquaculture.
Yes, actually, you're spot on. And you might know this, I'm not sure. So the Vibrio harvii that Bonnie always worked on got reclassified as Vibrio campbellii. So it turns out even that one is a shrimp infection agent. And they cause devastating disease. And there's 3 or 4 different species that do it. They can cause, I think it's like 8 to 10 different disease etiologies. The most well-known is called acute hepatopancreatic necrosis disease. And it kills the shrimp in massive batches, and it's causing a lot of problems in aquaculture. It has been for a while.
So that means that if you were looking at the brine shrimp system we were discussing before, that makes perfect sense.
Yes.
Because that's also a crustacean.
You got it. And so we use the brine shrimp as test models in our lab because we can do it in 96 well plates, and it's very convenient. We also collaborate with the University of Arizona in their aquaculture pathology lab, where they study the big shrimp, the ones that we eat, so the penniad shrimp. And we have happily done some experiments with some of our inhibitors that we see a lot of evidence that they can protect the shrimp from disease. And so it's not curing human disease, but it is helping with possibly with our aquaculture and thinking about money and investment in economics and things like that. So, yeah.
And now I'm going to make you cringe. It used to be the way that they dealt with these particular diseases in shrimp aquaculture was to just dump in antibiotics.
Yeah, they actually, I think, I can't remember when that started getting really nixed. There's quite an important rule against using antibiotic treated shrimp. And that's not true for all of our, all of our aquaculture, but at least as far as I'm aware, I believe the US and the UK will not import shrimp that have been treated with antibiotics. So that is probably a good thing, but it does mean our shrimp farmers have very few choices in how to deal with massive disease outbreaks.
Until you came along.
Well, we hope. Ours is an interesting compound, I think, and there's other people, of course, that study quorum sensing inhibitors and think along these lines, but they're not antibiotics in the canonical sense. They don't stop growth or kill the bacteria. They are antibacterial disease, but they are not antibiotics in the canonical definition. So they're interesting things to think about as therapeutics for disease that are broadly, we can think about that with any quorum sensing bacterium, really.
Mm-hmm. No, I think it's a lot better than dumping antibiotics. I often thought that phage therapy in aquaculture would be, a good thing too. And I know there's some people working on that in Indonesia. So as people new to this kind of idea that you're talking about, I remember reading about shrimp diseases caused by some of these vibrios, vibriosis, in fact, it's called, and how now you're telling me that quorum sensing is involved with that. Could you kind of walk through what would happen? In other words, you have a wonderful model system with brine shrimp, and then you do what to it in the microtiter wells?
Yeah, it's a great question. And yes, thank you for reminding me to say that the general term for disease that vibrios cause is vibriosis. Whether that's a shrimp, an oyster, a human, a fish, it doesn't really matter. We call it vibriosis. And it can have a lot of etiology in these different organisms. But we use the brine shrimp to model this. So we have lots of different genetic strains of Vibrios that we can put in and say, is, like you said, a reductionist approach. Is this gene important or not? One could also imagine that, there could be ways to develop targets for putative future antibiotics, right? Or even future anti-quorum sensing molecules that might be able to treat vibriosis. So we hypothesize as a field, right, that if you block quorum sensing and quorum sensing turns on the disease, that perhaps the disease could be lessened or eliminated by treatment with compounds. We've only just started.
So can I?
Yeah, go ahead.
Can I just amplify a little bit? I mean, folks sometimes aren't as clear on how to set up model systems as they might, and I appreciate that. So if you're dealing with a disease, say in aquaculture, with these good size shrimp, the ones that I like to eat with cocktail sauce, obviously you can't study them being diseased in a lab normally simply because of the logistics of doing so. So now you're using brine shrimp, and remember the name for brine shrimp for all of us growing up reading comic books were sea monkeys.
Correct.
So in any event, with these sea monkeys, you have a model system that's actually able to work with. So you add a little bit of, say, a dilution of a bacterium that you suspect is pathogenic, and then it kills the brine shrimp.
Mm-hmm.
Is that what you're saying?
Yeah, that's exactly right.
How do you?
Yeah, so
how do you measure them? How do you measure them being killed?
Sure. So this is something that is really fun to actually do, because you can see the brine shrimp they're big enough that you can see, small enough that they can fit in a 96 well. So like around the size or smaller than a dime, even a little culture well. And when the brine shrimp are happy and they're eating and they're just swimming around, you can see them shooting all around the little well. And if we add bacteria that will kill them, you will see very quickly their motion will slow down, and then they will fall to the bottom of the tube. If you look more closely with like a dissecting microscope or something, you'll see a lot of degradation, no movement. They're clearly dead. We've even seen some really funky things, and I don't think we're the first ones to see it or anything, but certain bacteria will just cover them in biofilms. They just become encrusted in biofilms, and it's kind of gross but fascinating. And so it's really easy to monitor the brine shrimp. And the big shrimp, I have not been to those facilities, but they fascinate me, and I'd love to go see them. They have huge tanks, so 20-liter, 100-liter tanks that are, they put the shrimp in and certain ratios, so different numbers of shrimp depending on the size. And they just, like you said, put in a dilution of a bacterial culture, and they monitor the shrimp, and they count how many are alive. And it's a little easier to see those guys and decide if they're alive or dead. Same idea, different scale.
I was going to say, it reminds me very much of how they do stuff with nematodes, Caenorhabditis.
Yes.
Where they let them feed on bacteria and then they poke them with the
yeah.
They poke them with a needle. Do they move?
Yep.
And I love that idea.
Yeah, it's really we're trying to figure out ways to scale it up. And one of the things my student has realized is that if you put them in like a conical tube, the dead ones will just fall to the bottom, and the live ones are just swimming around at the top. You can siphon off the ones at the top to just move them to another place. So it's a cool system. Even as we go more large scale with it.
Well, not for the shrimp, but it's still a cool system.
Yeah, I will say I have some trainees that they've said to me, the only reason they're okay with doing animal work on shrimp is they don't have a nervous system, so they're okay and they don't feel the pain. But I'm not really sure how that all. Yeah,
I understand.
Yes.
So, Julia, in previous episodes of Matters Microbial, we've discussed things that are adjacent to some of the ideas we've talked about today, like antibiotic resistance, like the concept of phage and how they attack bacteria. Do you think that there's a good connection between quorum sensing and these ideas?
Oh, absolutely. That's a really nice thing to bring up. So There have been studies in numerous bacteria now, not just Vibrio, of course, but Pseudomonas, off the top of my head. We know that in some of these systems, quorum sensing can regulate antibiotic resistance, and the presence of antibiotics can drive the quorum sensing response. So again, coming back to that idea of sociomicrobiology, which again, my colleague Josie Chandler studies, and this is an important area for her group. And likewise, we know that phages, both lytic and lysogenic, are impacted by quorum sensing regulated genes. So there have been studies, again, off the top of my head, that I've seen in Pseudomonas and in Vibrio that are showing genes that are regulated by quorum sensing impact the efficacy of phage infection or particle release in different ways. You can almost imagine if you were a bacteriophage, you would benefit from infecting a culture at high cell density or a community at high cell density, right? When there's a lot of bacteria around, a lot of targets. And then once that one cell released its progeny, there would be many, many, many more cells for those to infect. And so there's not surprisingly been some connections drawn there. And much like every other bacterial and bacteriophage war that we've studied in the field, I anticipate there are probably fitness impacts of quorum sensing on phage infection and bacterial defense. We also know that quorum sensing regulates bacterial defense, such as CRISPR. So it's absolutely all combined and regulated.
I have a final question for you about this, and honest to goodness, I'll bring it back to you. But here's my question for you. One of the things that I like to do for fun, I know I have a funny version of fun, is I like to show students that when they plate out from marine environments, they often get agar-degrading bacteria. Mm-hmm. That is to say, the bacteria are breaking down the agar that solidifies the media around them, mm-hmm. leading to a pit. So we make lots of jokes about Brad Pitt and pitter parties and things of that nature. But I'm wondering if quorum sensing's involved with that.
My guess is it probably is, if it's a Vibrio or even if it's another gram-negative, hunting around in the ocean.
So there are some of these for whom I have genome sequences.
Okay.
And I have not looked. So I may write to you to see what you think.
Yeah, absolutely. There's very well-known proteases that have targets that I could imagine would be, we know we have casein-degrading proteins and, hemolysins, and you pick your toxin, but there's many toxins encoded by these bacteria for sure.
Well, as we start to wind down a little bit here, I had one question. Where do you see your work in, say, 10 years' time, besides the Nobel Prize?
Oh, you're funny. That's an interesting thought. So, I tend to think more in 5-year chunks, right? That's like a grant period.
Yes.
So we're very interested in thinking about these host systems and mm-hmm. using the tools we've built. So the compounds, the synthetic autoinducers or quorum sensing molecules that we have, the different Vibrio species, and asking how that impacts inoculation and colonization and competition in these different host models. So that's one thing we're really interested in. And I have a new postdoc starting that's thinking about mixed communities and how Vibrio quorum sensing plays a role in that. In yet another system, the oyster. So it's an interesting concept to just we have these kinds of set host systems that we can ask very specific questions. And we have a lot of tools to do it. So that's one area for sure. It's hard to think 10 years, right. But we are very invested in testing these compounds as potential therapeutics. And part of our goals are also to use that tool to understand the biochemistry and the structure of our protein and how it functions inside the cell. Maybe we can develop even better compounds or more stable compounds or you name it. So those are things we're working on.
I think you've settled into a wonderful, groove sounds negative in a way, but I don't mean it that way.
No, I like that. It's a groove. It's a good one. Yeah.
Yeah. And what I like about it is that it's clearly productive. It clearly involves students at all different levels, ranging from undergraduates to people who were essentially colleagues of yours.
Sure.
So it's very inclusive, and it also has a known positive beyond the laboratory in a way that's surely going to get people really excited.
I hope so. We love what we do. I hope it keeps being positive that way. Yeah.
So if you won't mind my saying so, you certainly brighten the field.
Oh,
that's for you.
Thank you.
Luciferase wise, right? There you go. I don't know if you know this, by the way. Are you a fan of Doctor Who?
I'm not a huge. I know some about it, but I don't watch it.
My favorite moment was when I got to meet David Tennant, and my wife had done a Lex portrait of him.
Oh my goodness!
And I was able to talk about that with him.
And
he signed a photograph to me from one doctor to another, which made me very, very happy.
That's fantastic. I love it.
Yeah. So the point is, science is part of our lives that informs what we do all the time. You already mentioned working in Larry's lab as an undergraduate. What was the one observation that really sold you on being a microbiologist? There had to have been one thing.
Oh, yes. And it'll tie back, I think, to your chromobacterium. So my project in Larry's lab, I had this really unique one. There were two new professors that studied red-backed salamanders. And she had mentioned to Doc that it was well known in their field that the mother salamander, when she was on her eggs, protected them. But if the mother was taken away, the eggs would become covered in fungus very quickly. So we hypothesized that there were antifungals being made. And I spent some of the first days in the lab swabbing the stomachs of salamanders and isolating individual bacterial colonies. And we then tested them on agar plates that were filled with yeast, so that I think it was Candida. So we saw if there were zones of clearing, we thought they were making antifungals. And of course, we found a few. And we were in the process of isolating and identifying them. And those I can't even remember now, they're those kits where you like inoculate it and you stab it through. It's like in a hospital and you can get some very basic like information about what carbon sources, nitrogens, and things like that. Mm-hmm. And then you get a number and you get a code and then you can look it up. And the day that I figured out that we had a Pseudomonas strain, I remember running down the hall and I just interrupted Doc. I don't even know who he was talking to and couldn't contain myself. And I think both of us knew at that point I was sold. So that was it. I was a microbiologist and no turning back. So.
And that's our goal, is to create that. You've heard of a big tent? I'm going to call it a big Petri dish. And the idea of this is to make science accessible and available to everyone, because everyone brings something that's interesting into it.
Absolutely.
And you just have to have them put aside their, the disease I had in my 20s of trying to be cool, and just letting their enthusiasm really show. And that was really important. Look how it changed your life.
Absolutely.
Again, ditto for me when I was like 10 years old and I saw a Petri dish with glowing bacteria. I was sold.
Absolutely.
It's that kind of thing. It's been wonderful having you on the podcast, and I really appreciate your time and your enthusiasm. And I'm not even going to say it's been enlightening because that's too much of the light jokes, but it certainly has been. I really appreciate your time. It is such a pleasure to have you on this show.
Mark, it's always a wonderful time chatting with you. I'm so glad you invited me. And thank you so much for taking the time to talk with us and to bring this to the community, bring quorum sensing out even more than it already is.
Bacteria don't exist as individual cells, floating around a 2059 tube, nor are they on Petri dishes. They're part of a large consortium.
I agree.
And there we go.
All right.
My very, very best wishes to you and your family.
You too, Mark.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes, with tasty links as always, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Julia Van Kessel is Associate Professor of Biology at Indiana University in Bloomington, Indiana. Many, many thanks to David Renata for superb editing, and Reber Clark, as always, for the wonderfully quirky music. I hope you've enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.